So we rearranged a little bit, and I decided I'd rewrite my talk really quick after Jim's because you guys had so many awesome questions that I thought we'd show you some of the work that we're doing to answer some of them, and hopefully keep inspiring ideas for hackathon projects. So I also have a background. My training is in clinical psychology, and my idea was to like study functional impairment and pathology and do computational models of it, which is now kind of turned into computational psychiatry, and that made it a lot more boring when everybody else started doing it. So we had to move on. So I'm always in trepidation when people ask what I study. And we had a recent lab discussion where we were like, what should we put on our website? Because it's essentially what I put in my job statement, and I've never updated it from what we actually do. And so broadly, it's something at the interface between cognition, so like learning, memory, decision-making, and emotion, and then all kind of under the umbrella of social stuff. So does that answer the question? OK, but today I'm really just going to focus on our work on basically essentially one study that we're working on to try to understand emotion. So 90% of all research on emotion in the brain, if not 99, is basically this. You look at a picture, and this is what I think they're asking. What is this? What feeling in this photo is this photo intended to convey? What do you guys think? Good, probably, yeah. But how do you actually feel? Probably not great, although it depends what mood you're in. It might change you a little bit. What about this one? Yeah, OK. Probably not good. And you probably doesn't actually change your current state, right? And so what's the problem here? I think there's so many to enumerate, right? So basically, I think exactly as Jim articulated, that a lot of research that we try to do strips out all the contextual information. And when you're studying emotion and social stuff, that is the key. That's the sauce that makes it happen. It makes us feel things. So that's obviously a problem. Obviously, how do we assess how you're feeling? It turns out that like a 5-point Likert scale is probably like a pretty impoverished view of like what your internal state is at the moment, hopefully. So that's obviously another problem. Right, what was I going to say? OK, so I'm just going to give you some examples that maybe will evoke different feelings. Sorry. So you guys might have remembered that this happened. It wasn't a bad dream. Donald Trump was elected president, and a stunning repudiation of the establishment. This was the New York Times headline that next day. And half of the country or less was ecstatic about what this meant for the progress and the future of our society. And the other half was terrified. And there's tears, fear. My class the day afterwards, we had to do like a therapy session because everyone was crying. And so let me just, sorry, I can't get the sound to work, so I'm going to use this mic really quick. OK, and everyone was kind of worried about different things, but it wasn't really so bad.
The sharing of
false news has become all too common on social media. More alarming, some media outlets without checking facts first. Unfortunately, some members of the media use their platforms to push their own personal bias and agenda to control countries. Exactly what people think, and this is extremely dangerous to our democracy.
So luckily, we have lots of independent thought left, and not state-sponsored media and news and all of these things. None of those were like legitimate fears. So really, all I wanted to say about this was all emotion paradigms assume that everybody has the exact same experience. We're all going to be synchronized in response. And of course, that's preposterous because none of you guys probably felt the same way as other people felt when they were hearing this on the local news, right? So there's this bifurcation in our feelings, and that comes from our internal states, our goals, and our past experiences. And those are all things that you remove from when you decontextualize your stimuli. All right, well, actually, I don't need to ask that. OK, so controlled experiments decontextualize psychological processes. They also require an explicit model of task structure. And so when you're starting to create these experiences, that might be hard because you don't know what people are going to do or how they're going to feel or when. And they also assume that everybody has the same experience. So hopefully now it's like a little obvious why that might be a problem. And I'm just using emotion, but I think that probably extends more broadly. So we can think of emotional experience as an endogenous process. So Exogenous processes might be things that are trying to relate to understand the external world. So our sensory systems, they're going to be responding really quickly, and that likely is probably going to be more synchronized across people, as Alex was suggesting earlier in his comments. But other regions that might be integrating lots of information at the same time, it might be happening in a much slower time scale. And it's probably nonlinear with respect to the outside world, and it's basically going to be having to integrate these goals and internal states, so brainstem and our prefrontal cortex and our hippocampi and all these different regions to say how we're feeling. And goals, obviously, of what we're trying to do are going to make a big contribution. And so I think there's kind of two predictions we might, and also we think that people might be varying. So even how you are at one moment in time might not be the same if you in another moment in time. So I like to think if I look back in my past life that I thought the same way about everything as I do at this moment, but obviously that wasn't true. That's why I like looking at high school or middle school photos are so embarrassing. Like, did I really think that? Did I wear that? Did I really think that was cool or listen to that type of music? So we think that the regions that are involved in these emotional experiences, because they're like integrating all this information, they're going to have like a long temporal receptive window, and this is what Chris Honey is going to talk about from his work that he's done with Uri. And we also expect that there would be a lot of heterogeneity across people because we all have different experiences until we start getting the same news information and other things. Okay, so the vmPFC is interesting because it has a unique pattern of functional connectivity. So this is work that we did a while ago on parcellations of the orbital frontal cortex. And even within the medial part of the ventral OFC, there's divisions that seem to be connected differentially. So this, and the kind of like the medial wall, that seems to be more of this default network, where all just slightly laterally, it correlates with other regions, which I not remember which one it is. Oh, down here. So with the kind of ventral pallidum, and then negatively correlated with the VTA and the PCC. Some other work we've done, so this is using resting state connectivity, but we've also looked at it using meta-analyses. So this is about over 10,000 published studies. We take all the coordinates, and then we can do the same type of analysis where we look at patterns of coactivation in published work. And in the medial part of the PFC, there's kind of these three broad subdivisions that seem to be distinct on how they're connected with the rest of the brain. And they also, we can do this type of reverse inference where we can figure out what functions are likely ascribed to them based on what people are talking about in their papers. And they also correspond where they seem to be distinct functional connectivity profiles. And we can go more fine-grained to dial it up. And even in the ventral medial prefrontal cortex, there seems to be different from like the dorsal medial, for example, where one might be more associated with processing social stuff, or the other one might be more with reward, at least as if you use what people describe it in papers. We also know that the vmPFC is probably involved somehow in affective appraisal. So there's a really large body of research studying value representations and anticipation. There's also work on placebos where those types of things might modulate your experiences. And then also in self-generated emotions, you consistently see the vmPFC. Okay, so the approach that we kind of take is a couple things. So one, we think that the pattern of activation conveys some type of information. So there's some spatial representation of psychological states. So this whole idea of even using decoding from published work. So we can say, so given this map, if you guys who read a lot of these types of papers might say, oh, well, those regions probably have something to do with emotion because you've read like thousands of papers on this. And our algorithms that read papers can say the same thing. This is with a probability they think that any of these functions broadly might be in the presence of this configuration of spatial activity. We can also, so this is like using resting state or like meta-analysis stuff, but we can also specifically make a map to probe a type of emotional response. This is just negative affect. And so we call this the PINES, the picture-induced negative emotion signature, where we can train a multivariate representation of that can predict how bad someone's feeling at a given moment. And then what's nice about having this type of model is we can do model comparison. So we can see how does this model compare to another model in predicting something. What about if we use nested models? So can we figure out which regions of this model seem to be ones that contribute most to the explanatory power? We've also done some work which I don't think speaks to the quality of it, but it's been under review for 4 years. Mostly, I think, confusion and laziness on our part to get it in. But this was another idea where we try to look at gene activation or gene expression from data from the Allen brain atlas, and then tried to see if we could decode what the genes might be doing based on their spatial configuration using neurosynth. This actually was a project that came out of a hackathon, so which is kind of what the whole week's about. Okay, so I think as Jim really nicely stated, we can think about information as a point in a multidimensional space. And so this is using Jeremy's hyper toolbox. And these are vmPFC patterns while people are watching a movie. And each line is basically a subject. And it's basically trying to say, are they going in the same direction at the same moment in time? And as you guys have already started noticing in some of Jim's plots, that the hyperalignment stuff seems to really help in certain regions, but specifically in the ventral medial prefrontal cortex, it doesn't. And part of that might be due to the fact of the susceptibility artifact, as some of you guys were indicating, or it could be because this is the most idiosyncratic region of the brain, because it's the one part where it's unique of the experience it's constructing is unique to us and our prior experiences in the world. Okay, so in a hope to try to study this, I'm going to tell you a little about this study. So this was Eshin Jolly and Jin Chang, who's at another brain hack week in Seattle. So they basically collected a whole bunch of different data sets on watching the show Friday Night Lights. So as Rob mentioned, maybe if we have stimuli, unlike Indiana Jones, which has more social stuff or more emotional stuff, that we might have more variation in these regions that might help with hyperalignment or probing these functions. And so this particular show, I'm just going to show you just a work from the first episode, but it was kind of designed, it's one of these like ABC family dramas. It's supposed to like have lots of character changes, dynamics, and lots of affect. So the question is, do people generate emotions in the same way? So this is basically plotting temporal intersubject synchrony, so how much people share the same time course in each of these regions. So in V1, and you see each line here is a subject, so this is going through time. And you can see there's a high degree of temporal synchronization, whereas the medial prefrontal cortex has low. And I think this is partly why the hyperalignment stuff it's reflecting kind of the same type of signal. So I don't think this is terribly surprising. And if we take each subject and the time course and then look at the pairwise, so every kind of column and row here is a different subject, here we see there's overall, on average, a high degree of synchronization, where in the MPFC we see a really low, so it's effectively centered at 0. Okay, so what about if we try some type of functional alignment like hyperalignment or shared response model? There's a whole bunch of different versions of it. So this was in one study we did. This was only 13 subjects while they watched this first episode. This is basically the temporal intersubject connectivity after hyperalignment. And I think this nicely follows exactly all of the prior work that people have done. So we replicate all of that, where you get really nice functional synchronization across people, but notably absent is the vmPFC. And then like, okay, well, let's do this in another study. So this is a bigger one. This has 35 people. And I kid you not, this is different data, but the reliability, if you take the spatial correlation between these, is 0.99. So it's like a really robust finding that Uri's lab has been doing for like the past 15 years. We can also do spatial intersubject connectivity. So we can say, well, maybe we don't want to average in a region over time, but maybe we can look at every TR or every snapshot and say, what's the spatial pattern? How similar is that across people? And then basically I'm plotting two. So the lighter colors are the anatomically aligned, and then the darker ones are functionally aligned. And you can see in regions like early visual that you get a dramatic boost in even the spatial thing, so that the hyperalignment seems to be working as you'd expect. But in vmPFC, you see absolutely no benefit in this particular data from that. Okay, so if we think of as information being processed in this kind of spatial vector, we can, I might not be able to say like how is information processed and what's the algorithm that it's doing, or can I even figure out what the information is, except with respect to any other kind of state that you could be in. So here I'm plotting, if you take that vector and you correlate it with every other moment in time, this is just for a single subject. So this is what we call like a temporal recurrence of a spatial pattern. And so basically I'm showing here is time, and this is over 45 minutes. And I think what's really striking to me is that you see that there will be patterns that persist for minutes. So this is like about 3 minutes, like this block right here. And so I think this type of time scale, people really haven't looked at fMRI that well. So in resting state, people do, but they don't know what's going on, so they can't ever quantify the response for the duration of time. And then the other thing is, it looks reasonably low dimensional in the sense that there are different states that are happening, but there's not too many of them, and they seem to be kind of anti-correlated. Okay, so the next thing is like, okay, well, this is interesting, but how do you know this isn't noise? And one way we try to do that, of course, is by saying, well, if it's noise, then it should generalize beyond people. So if we look at different subjects, the same type of plot across them, they're all very, very, very different. And I think this is partly what's been contributing to this issue. And so I still don't know if this is noise or not. But there are some time points where different people might be having a similar thing going on, but not all of them. Okay, so different ways to quantify that. We can take the mean over all of them and then do like a t-test to see like what regions seem to be consistent across people. And this is FDR corrected, so there's like nothing really other than like a couple seconds that is probably just reflecting temporal autocorrelation. We can take the lower triangle of this and say, what if we look at the similarity of this spatial temporal pattern across people? And sorry, I can give you a color bar. So this is 0, and that's what we see here. So there's essentially no similarity of these triangles across people. But it's not true of all of our data. So if we look at early visual again, this one, we see lots of structure that's consistent across this kind of spatial temporal similarity. Okay, so the next, one of the hypotheses we're trying to test is like, does it actually have a long tempororeceptive window? So we probed the temporal autocorrelation of basically all the regions in the brain. So this is, well, okay, what do I want to say? So we basically subdivide the brain into 50 different parcels, and then we fit an exponential function to basically their autocorrelation function for every subject. And so I'm plotting all the subject lines, and I'm plotting 2 separate lines for the V1 and vmPFC. And you can't see that there's 2, but because they're on top of each other, because they're very similar with the spatial autocorrelation is, which is good. There is variability across the brain, but it's not huge. But if we look at the temporal autocorrelation of the spatial pattern, which isn't just the mean voxel activation, it's something about the configuration of voxel responses, then we start to see that the vmPFC actually has a much longer one, and among other regions in the brain too. So what's plotted here is the time it takes to get to 0.1 here. So this is actually in TR, so that's about 22 seconds it takes. You're still having a bit of autocorrelation at 22 seconds in the spatial function, on average. And each line here, again, is a subject, so you can see it's really reliable across people. Okay, so the next thing we want to do is we saw that structure in the recurrence matrix, but we want to basically try to identify time points. So looking at the whole thing doesn't seem to be working, but maybe if we narrow into discrete moments in time, that'll help. So to do that, we assumed that there's some latent state changes that this activation profile and the vmPFC might be manifesting or reflecting, and we used a hidden Markov model to try to identify when these state changes happen for each subject. So we have a matrix that's basically voxels in the vmPFC by time. We did a PCA on this just to make it orthogonal for our hidden Markov model, and then we're trying to estimate what states they're in and what the transition probability is. And this, we can run the model back through the data and then get a prediction for, given a moment in time, what the model thinks that subject state's in. So next, since we do this for every single subject, we get a prediction for what the states are, but we can try to align all of the predicted states across subjects using, essentially, we just used probably a crummier version of hyperalignment, where we're iteratively going through and trying to align each subject. And so what I'm plotting, every color here is basically a state, and then every row is a subject's predicted state from their model. And so their models are interesting because it allows every person to have a different spatial configuration, but now we can start seeing if we all have idiosyncratic representations, but maybe they're aligning in time at the same moment. And that's basically what we're seeing here. So you can kind of read this like a rastergram, where if you take the mean, we can kind of see the aggregate over people in the sample. Okay, so the question is, do people change states at the same time? And that last plot indicated that seems to be happening. And then we can map this back onto the movie, which is, I think, unlike in resting state stuff, where we don't actually know what the internal state of somebody is. We still don't know when they're watching a movie either, but we can try to see what types of things are going on in the movie at the time. And this is not showing up terribly well, but some of these peaks end up being key narrative elements in the movie, and also, in this particular episode, the most emotionally arousing ones of them as well. And you're like, okay, well, that's great, you did all this. You have a lot of degrees of freedom here, and how you're doing all your analyses. How robust is this? So this is with 13 subjects in the first study on a different scanner. On a Philips scanner, this is on our new Siemens one, 35 subjects, and we basically see, it's not perfect, but all of these peaks end up seeming to be fairly reliable across these two different samples. So that's cool. So there's something happening in these state changes that we can detect reliably across people if we start relaxing assumptions of how it's being represented in the brain. Okay, we should have stopped there, but it's like this burning question, like what actually are these states reflecting? Is it narrative elements? Is it emotion? Is it just a scene change? Like what's going on? Well, it turns out that's hard to do, and I'm just going to caveat this and say I don't hope to convince you, but I just want to provide some ideas that might trigger some thoughts for maybe your hackathon stuff. So one way we can do it is, well, let's look at some behavioral expressions of what might be happening here, some objective things that aren't simply self-report. So in our lab, we've built these headsets to be able to record facial expressions. They use GoPros, so we can do about 120 hertz sampling frequency. And then Jin here has kind of led efforts in development of this and written a software toolbox to basically allow synchronization so we can run social experiments or all types of stuff with these headsets. So this is just an example of what the recordings look like. So some people are like, well, people don't emote when they're by themselves watching a show, which obviously isn't true. Some people are very exuberant. And some people are flatter, right? So there's lots of variation in this. So we can do the same analysis. This is a different sample now with just people who are outside the scanner with their facial expressions and say, are people changing states in their facial expressions at the same time? And it's a little slower and cruder, and there's a lot of like parameters that we can make this look a lot better. But if we use the same method of how we derived it from this, we basically find a lower number of states, so it looks a little like it's even slower time scale, but they're lining up at the same key scene. So this is that scene that I was just showing you where they're like, it's this winning touchdown by the quarterback, the backup quarterback, but the only reason why that's important is because right before that, the star quarterback, the QB1, as they say in the show, I don't know much about football, but I know the quarterback throws the ball. He basically was paralyzed from a bad hit right earlier, and so everybody thought the season was over. It's really sad, and that's like obviously another peak that's happening here, and the facial expressions seem to be reflecting that too. So I still don't know if it's and I don't even think it's important to distinguish between the narrative parts versus the emotion, but clearly this has something to do with the generation of affect. Okay, and then we can also, aside from these objective things, we can just ask them, but it turns out that that's hard to do when you're watching these movies, because you're like, okay, well, how are you feeling? Well, let's just have you rate how you're feeling over the whole time scale, or the whole show. And you're like, okay, but that's only one dimension. I want to know a whole bunch more, so let's do it again. Why don't you watch the show again and rate how you're feeling? Now do it again, watch it, you do it over and over and over again, and you can imagine that the emotional experience has now changed a lot, considerably every time you view it. So it's kind of a tricky methodological problem. So how we are trying to solve this is doing like what basically is really popular in the tech industry. So it's using collaborative filtering algorithms, which are kind of the crude engine that drives like Spotify and Netflix and Amazon, how it finds our preferences. So what we do, so every row here is a subject, and this is, I don't know, like a couple hundred subjects, and we sparsely sample how they're feeling randomly. So we stop the movie, I think like maybe 12 or 13 times for this 45-minute thing, and then we ask them to rate how they're feeling, if anything, on any of these 16 dimensions. And we do that for all these people, and then we can use these collaborative filtering algorithms to basically reconstruct for each individual how they were likely feeling, given their similarity to everybody else. And this is the reconstructed time course, and if you were imagining the mean, so this is joy and this is sadness, it basically corresponds to those key moments in the movie where they're all really excited and happy versus they're sad that the star quarterback just got injured. Okay, so if we do the same analysis on this hidden Markov model on that, we also see that it pulls out the same states. So there seems to be valence here. We're seeing emotion. But none of this, we've actually made any claims about the specific feeling they're in. We've allowed that to vary across people. We can try to see like how consistent is this across all these different measures. So these multiple studies, these multiple modalities of probing emotion. So this is like a cross-modality consensus. So we just look at the similarity and the time courses for all the average states across them, and we can cluster them and find that there are some of them that seem to be consistent, that transcend the type of modality that we're probing. And they, again, this blue one is the really happy scene, although it's a little flat right here. That's that one. This pink one is the one where he gets hit and breaks his spine. And this is another one where there's a bunch of jokes at a party and it's very ambiguous and people have different responses. I could go on and on and on. We've tried to manipulate how, if you change goal relevance, how that might change synchronization. Let's see, I'm just going to say one thing about this. This is led by Christina Rapuanu for her dissertation, where she had people who were like basically undergrad alcoholics. Turns out there's a couple at Dartmouth. And she scanned them right before Friday night before they go out, and then right before they go to class on Monday mornings. So it's basically trying to do a goal manipulation. And then she didn't do the Friday Night Lights manipulation. She basically showed them pick different types of videos. Some of them involved alcohol, and essentially finds that when people are on Monday morning going in, that she replicates the same effect that we just saw, where there's very little synchronization across people. But when the goals are synchronized, when they want to go out, she sees increased synchronization in the vmPFC when it's goal relevant, and it turns out it's specifically in the alcohol videos, which is pretty interesting. Okay, so right there, I'm just going to stop and thank Eshin and Jin and pretty much our whole lab as a group effort to be able to collect this data and analyze it. So, thanks. Yeah, Linda. The secret sauce. I'd tell you, but I'd have to kill you. So I looked until it supported my hypothesis. That's what I did most of the time. No, so how we did it, it turns out it's really hard to do for this because you have to make some assumptions to be able to do it. So the one that we made that I probably don't really believe, but you have to have at least some assumption, is that every subject has the same number of states. So then what we did is it's essentially like a split half reliability to find consensus. So we divide the sample up randomly. We have them go through and we iterate through the alignment across subjects. And then we do that for the two halves and say, at which number of states do the two halves seem to be most in agreement? And I'm just going to tell you right now, it's super noisy. So if I run it multiple times, it doesn't always converge on the same time. Usually the peaks are the same, but the maximum changes a little bit. But yeah. Other questions? Yeah.
Do you require any trait level measures on subjects at all? So just sort of baseline emotional processing ability, or something vanilla emotion tasks that's helping people still measure a range of performance on?
Yeah, it's a good question. So I'm gonna guess no, mostly because when we were starting, we had 13 subjects, and it was like, okay, well, we could do it, but then, and they were already so in that one we had them come in for 4 episodes, so it was I don't know, 6 hours, and it was just to add another hour of all the questionnaires for not a lot amount of time. The other thing too, yeah, I think it's fair to be provocative here. To what degree do people, do you think, have good insight into their emotional feelings?
Yeah, well, so self-report is one thing, but even just some of those reading the mind and the eyes.
Yeah.
Like that.
Yeah, so you're like, okay, well, maybe self-report, but then we can have these other questions, but then how are those validated? And almost all of them, mine eyes ones, maybe, but almost all of them are validated on other questionnaires, so it's this circular thing where no one's actually validating behavior. Yeah.
It would just be interesting to see if the subjects that were more similar neurally were also more similar on some totally independent.
Totally. Yeah. So we don't have questionnaires, but we have done a lot of work. So a lot of our other papers right now we're writing are on this idea. So if you take the RSA but now you do it on subjects, so how similar are different subjects and how similar are they on some other type of measure? So the ones we've been doing a lot are on impressions of different characters or their beliefs about the show or different things like that. So it's specific to this context, but then it turns out that the alignment on those types of things maps on regions that seem to be processing things related to it onto it. So we'll call it intersubject RSA for lack of imagination, but but I think that's a pretty powerful method. And so Andy's been doing stuff, Jin, and we've also been doing it on facial expressions, and it's pretty cool. You can actually, as a validation, how much do you like the characters seems to map onto when they're smiling at the same moments in time. And then we've also been doing stuff with just facial expressions alone, where if you do dyads or single people, the dyads seem to synchronize more with each other, and they seem to align their impressions more afterwards, which is interesting. Yeah, but I'm basically just trying to evade your question that I didn't actually forget to answer with them, but yeah, yeah. Yeah.
So I guess it's sort of like a slightly different question. It sort of came to mind also in Alex was asking from the previous topic. So you're talking about MPFC, like this level of variability. It sort of suggests there's something like almost categorically different about what it's doing, right? It's
Mm-hmm.
about internal state, which sounds really yeah, definitely. But I'm sort of curious, like how do you differentiate? Like you can think of an alternative, which is sort of like there's a cumulative difference, right?
Mm-hmm.
So something about V1 is maybe more similar. There's just less, think of it, fewer synapses for there to be variability. Like every single one is like slightly shifted in space and so on. And by the time you get to the frontal cortex, you just like, this anatomical drift is just too much really
Yeah.
to see the similarity just by that sort of realignment. So I guess the question is like, how do you differentiate? How do you decide when there's something different happening versus something like that?
Yeah, exactly. It's a really cool question. So I hope Chris will talk about some of the stuff he's been doing on that, but he's done work where showing that even the distance of how far things are, it's going to have different time scales, and and I think you're right that the more further you are in the processing stream away from things, the more, especially if there's different paths and all these different things are happening at different times, there's more noise accumulation that could be happening that could be driving it as well. And I think to be able to test that, I don't even know where to begin, but it's a really cool idea to think about how to probe more.
Maybe that's what this sort of whoever you refer to is doing, but maybe you could look for some sort of like you'd expect like a continuous gradient in the accumulated difference case as opposed to some sort of
Yeah, so we varied it a little bit, like to try to account for some of the misalignment across people. We did like a little bit of smoothing too, which helped. So we might be kind of missing some of that variation at the moment. Yeah, Andy?
I was wondering about the role of behavior in all of this, right? Because most of what Jim talked about, I think, and all of what you talked about is just passive viewing of the
Mhm.
situations, right? People aren't actually doing anything or engaging with real world scenarios. And I wonder
Right.
if you think this is an area that's like really colder and maybe behavior really matters.
Yeah, it's a great question. So I think it's similar to like what Jim said about there's no sensory motor stuff because you're passively laying in the scanner. Similarly, when you're watching a movie, you're not really. I mean, there's some kind of broad goal, which is to like not fall asleep or to watch a thing, or But yeah, it might be totally different when you're actually engaged in tasks or navigating the world. So I don't know if we want to share this at Hackathon, but we've gotten some rat data that has some MPFC recordings where they're just basically like continuous recording for like an hour, and then we're trying to use these same techniques to see if you can predict when they initiate a goal to go feed or get water or something. Yeah, but it's a really cool question. And also really important, I think, when you're designing experiments of like how stupid it is to try to study social stuff when you put one person in a scanner.
I was curious if you tried training them on, like, say, a different area that might encode, like, reward prediction errors, for example. So, like, would that give you, like, a different kind of, like, state transition thing?
Yeah, it's a good question. So if we trained it on different regions or to predict different things, what would happen? So I've run it on all of the regions. We're like writing this paper right now. Every time we get a draft, we're like, oh, this seems obvious. We should fix it, and that's one of them. And I got to say, I haven't gone through and summarized it yet for every region, but we've run it. I just don't have an answer for you yet. Yeah, that's a good question. Yeah, Ariel.
Double click on the claim you did in the introduction regarding the use of naturalistic stimuli. The negative stimuli that you showed the picture, it moved me.
Yeah.
And I'm sure it moved other people as well. And IAP's pool of picture, like the bad one, the negative one, it raises people's heart rate, people react to it, some people refuse to look. No.
Yeah. I spared you guys the morning ones. I didn't put any gruesome ones up for that. I didn't put any of those ones up for your basically benefit. But yeah, there are ones that can elicit a response, like in terms of arousal or something that's aversive. But is that the type of a response you feel normally throughout your day? the types of emotions that you are feeling. Like, so the first one you said, which is about the old couple, I think that one for sure could. And my guess is like there was huge variability across people. Some people might have had like their parents or their grandparents recently passed away, or they watched this go through, and that they might be more touched or relate to it more. But that's specifically, that variability becomes because you have a unique experience, I think.
But this is true for every stimuli. There is always an interaction between person history
Yeah.
and the reaction to to the external stimuli.
Yep.
So my question is, what is exactly that so it's not that unnaturalistic stimuli are not powerful enough or not engaging.
Yep.
Right? So this is not a claim. So what I'm asking is, what do we gain by using movies in comparison to using static?
So yeah, so I'll try to answer it in different ways. So I'm not claiming that they're not powerful enough or reliable enough. That's been shown. So the IAPS, there's a meta-analysis of all these different emotional elicitation things, and they are the most reliable, and they have an effect size like in the medium order. I think it's like 0.6 or 0.7 on average across like thousands of studies. But the question is if we're trying to study emotion, is that really emotion that we're studying by watching it, by looking at these clips that are being elicited? And so what do I mean by that? So it turns out that like all of the arguments in the field of emotion, which I personally don't find terribly interesting, not the field of emotion, but the arguments, they're like, is it a 5-dimensional model, categoric, discrete categories, or is it this like lower dimensional, 2-dimensional thing, right? And it turns out all of those things can be completely explained by measurement issues of how they did it or how they elicited it. And I think what has been underappreciated is if you just show a couple of these pictures and you ask them to rate on this like stupid scale that we treat linearly, but it's really ordinal, and there's all these other problems, and people even make emotional judgments about introspecting their internal states, that biases you to find a low-dimensional solution, and that's not actually what emotions experiences are. And so what we're basically doing is just sidestepping that. So we're saying, we know people spend, it's a billion-dollar industry, like the film industry, to craft narratives that touch people, and that they might speak to people in different ways at different moments in time. So we're just kind of leveraging, rather than trying to like, I know in Uri's lab you guys actually are constructing your own stimuli to do this, but we don't really have the funds to do that yet, so we're kind of relying on that. But the type of experiences we have, those I think are richer, and they might not just be like a 7 on a joy scale or something. It might be something else that we can be capturing.
But the fact that they are so hard to generate is, like you said before, you need very good directors and very good screenwriters.
Well, I don't know. So that remains to be seen. So another thing we're doing, that some of the stuff is from this grant, is we're using Moss stuff similar to what Alex did for his narrative stuff, except we're specifically interested in like selecting stories that are more about their narratives about them. And yeah, they're good stories, right? That's how they get there and they touch people. But so I think the big advance of using these naturalistic stuff as opposed to just like a static stimuli is that it provides contextual information. And so and these are studies we proposed in our grant, but if you show the same scene that's arousing, so that like in IAPS perspective, it shouldn't matter one way or the other, but now if you show the scene with all the context for why that's important, you should get a much increased response. And it might not even be increased, it might be fundamentally different too, as from working in your guys' lab would suggest that too. So. Yeah, but these are really good questions to probe and think about. And what I like is we should be skeptical about everything and not take like the status quo for granted about how we study stuff. So even because people have been using IAPS for the last 50 years or whatever, 30 years, that doesn't mean it was a good way to do it. It doesn't mean it wasn't a bad way either. Should we move? Oh, yeah, one thing while Jimmy gets set up. Yeah, come on. Yeah, come on. It's up to you. I think we might want to press ahead just for time.
All right, we're going to forge ahead.
But if you want to switch.